All right, and have arrived at our last functional group, which is the amino acid functional group. Now, the primary characteristic of the amino acids is that they are the building blocks, the structural monomers of proteins. Amino acids are going to be the structural monomer of proteins. They're primarily made of hydrogen, carbon, nitrogen, oxygen, and sometimes sulfur in very specific circumstances. But you're always going to have hydrogen, carbon, nitrogen, and oxygen in these structures. So I want you to take a look at this picture on the right-hand side. This R group is going to represent, this R group right here is going to represent a variable structure, a variable structure. So the same way that we talked about R groups when we were talking about functional groups, R just means that there is a variable structure. This R group is going to change depending on what amino acid you are looking at, right? It changes depending on what amino acid you are looking at. So in this case, the structure that we're looking at here, it is uncharged, but hopefully you can notice that it's supposed to be acidic. Well, we know that this is a carboxylic acid group, and this carboxylic acid group is going to have the potential to donate a proton. Interestingly, this is what we've talked about in class a little bit as well. This structure on the left-hand side, the hydrogen and the nitrogen, this is going to have the potential to accept a hydrogen. So overall, this structure is going to both donate a hydrogen and accept a hydrogen. So it is technically acidic, but its effect on a solution is going to be overall neutral because it has this capability of both accepting a hydrogen and releasing a hydrogen into its environment, right? So that goes into the 4 components or the 5 components of an amino acid. 5 components of an amino acid as labeled here. We have the hydrogen. There's going to be a hydrogen, an amino group, which in this case, we've seen what happens after the explosion, right? After the chemical reaction has occurred. So in this picture, we saw the chemical reaction or the molecule before the chemical reaction occurred. This shows after the chemical reaction occurred. You've got the amino group, the carboxyl group, which is going to be our basic, the amino group is going to be our basic group. The carboxyl group is going to be our acidic group. And then the R group is going to be our variant group. The 5th characteristic of an amino acid is this central carbon. It's going to be really important for you to be able to identify where the central carbon is when analyzing amino acid structures. There are 2 sort of essential categories of amino acid side chains, right? These R groups. So if we go back, if we think about our original structure, we've got the central carbon, you've got the carboxyl group, you've got the nitrogen group. I'm just going to give it a little plus there. You've got the hydrogen, and then you've got this R group. Now, depending on the characteristics of this R group, it's going to change the overall characteristic of the amino acid because, as we said, it has the ability to donate a proton to the solution and accept a proton from the solution. So the defining characteristic is really going to be this R group. That's the defining characteristic of an amino acid. Oh, don't worry about this label here. This is not talking about enzymes. We're talking about amino acids. Sorry, that's a label from a while ago. So the 2 categories we have are nonpolar and polar side chains. You guys know what the difference is between nonpolar and polar at this point. You guys should be able to identify, okay, a nonpolar substance is going to be something that does not have a high difference in electronegativity, whereas a polar molecule is going to have a high difference in electronegativity, which we can see here. You've got 2 highly electronegative atoms attached to a carbon chain. You've got oxygen and then nitrogen attached to this carbon chain. And because this carbon chain is so short, this this polarity is actually going to have a lot bigger effect on the molecule overall. So nonpolar molecules are going to have usually high amounts of carbon and hydrogen, and usually in the form of long chains or ring structures. Polar molecules are going to be charged or highly electronegative side chains. So the same way that we saw a charge occur when we were looking at carboxylic acids, right? We saw this negative charge occur. You're going to see the same situation with our amino acid side chains. So these are the the non-polar side chains. These are the naturally occurring amino acids that we are going to be worried about. Again, you don't have to memorize these structures. You do not have to memorize the amino acids. Some teachers will make you memorize the amino acids and whether or not they're polar. I am not going to make you memorize them. However, you will need to be able to understand their overall structure. So that's the 5 components that we talked about just a couple slides ago. And you're going to need to be able to recognize when a side chain is either polar or non-polar, right? So as you can see with all of these structures, there is mostly carbon and hydrogen, which indicates non-polarity. You do have a little bit of sulfur. You got a sulfur atom here, you got a sulfur atom here. But when you look at the structure of these side chains, these sulfurs and nitrogens are embedded within highly non-polar regions. So the reason these structures are considered non-polar is because they are embedded within highly non-polar side chains. If you were to add any more electronegative atoms to these side chains, it would completely change their characteristic and they may no longer be considered non-polar. They might in fact be considered polar, which is what you're going to see with the polar side chains. So what you'll notice with these side chains is that they all have some sort of highly electronegative atom in an accessible location. So that's the key thing here. You have highly electronegative atoms here and here with the sulfur and the nitrogen, but these highly electronegative atoms are not easily accessible. Sulfur is embedded between 2 very stable carbon groups. You've got a methyl and a carbon chain, which are going to really, really increase the stability of this molecule. Then you've got nitrogen, which is embedded in a ring structure, which ring structures are almost impossible to break in a natural structure. They're not impossible to break, but they're very, very difficult to break in a natural biological phenomenon. So what you'll notice about the polar side chains is that all of these polar structures are easily accessible. All of these polar structures, they're not embedded within a ring structure. They're not locked between 2 carbon chains. They're easily accessible. And so that is going to contribute to the to the polarity of these structures. It's going to make that electronegativity, it's going to have a stronger effect because they're sort of out there on their own. If you want to look into the deep chemistry magic behind this, I encourage you to do so, but you don't need to know it for AP Bio, but it can be helpful for understanding why these are considered polar versus non-polar. But if you don't want to look into the deep chemistry magic, just remember these are polar because they're on the end of a chain versus being on the center of a chain. Now, polar side chains actually have 3 different types. You can categorize them as either neutral, acidic, or basic. We looked at the neutral side chains on the last slide. These are our neutral side chains. These are still polar side chains with highly electronegative atoms, but they are not charged. They are not going to donate atoms to the solution, right? They're not going to donate protons to the solution. The way you can tell the difference is usually there's going to be a charge associated with acidic and basic amino acids. These are usually going to have a charge associated with them. So that's the examples that you see here. Aspartate or aspartic acid is going to donate protons to the solution. So again, it's like seeing a picture after a explosion. We're seeing the picture of the molecule after the chemical reaction has occurred and it has already donated its proton to the solution. The opposite is true for lysine. Lysine is going to be basic and it's going to attract hydrogen ions and it's going to remove hydrogen ions from the solution, right? Remember, when we're talking about acidic and basic side chains are going to be shown after they've removed or donated their protons to the solution. They're named because of the effect on their surroundings, not the actual amino acid. Remember, acids and bases are determined by their effect on their surroundings, by the effect on their surroundings. That's the key thing here. They are not determined by the amino acid or by the nature of the amino acid itself. Because if you were to look at aspartic acid, for example, you would say, oh, this molecule is lacking an extra hydrogen. This molecule in this state is lacking a hydrogen. So that means it's going to be basic. No. You got to be careful. It's about what they do to the solution. Determining an acid or a base is how they affect the overall solution or their surroundings. So these are all of our polar side chains. These are all of our polar side chains. We've got the neutral side chains, the acidic side chains, and the basic side chains. You'll notice there's only 2 acidic side chains and only 3 basic side chains. So that's important to keep in mind. If you need to, you can memorize which side chains are basic and which ones are acidic in order to help you when you're determining if something is either polar or acidic. It can be helpful to just memorize, okay, these ones are acidic, these ones are basic. If it's not acidic or basic, then it's going to be polar or nonpolar. Okay, now what we've been talking about here is just purely defining the characteristics of amino acids. We've given you the pieces that make up the amino acids, but now we need to talk about how these amino acids link up together. And this is going to be, again, through those hydrolysis and synthesis reactions. So what we've got here is a huge polypeptide. This is a string of amino acids. It's not yet considered a protein. It's not yet considered a protein, but it is a string of amino acids linked together via their carboxylic acid groups and their amino groups. So if we were to draw what this structure looked like without, being linked to other molecules, here we're looking at this guy right here, right? He would have the hydrogen. Here, let's make a little more space for it. He would have the carbon attached to the hydrogen. He would have the carboxylic acid group, which you'll notice in this image. The carboxylic acid has bonded, the oxygen is now gone, and it has instead bonded to a nitrogen. It's going to have the R group, which is that variable side chain group. And then you're going to have the nitrogen. Now, normally nitrogen has, or in the case of, after the chemical reaction has occurred, after they have donated or accepted their protons, this is what the molecule is going to look like. It's got that positive charge. I'm going to go ahead and erase that. It's got that positive charge because of the proton that's been attracted to the lone pairs of nitrogen. Now, what's going to happen is that this original molecule is going to be attracted to the nearest polypeptide or the nearest amino acid. It's going to be attracted to the positively charged nitrogen. You've got the positively charged nitrogen attached to a carbon that's got a hydrogen, that's got an R group, that's got a carboxylic acid. So what we're seeing here is another reaction in which this oxygen is going to be attracted to this hydrogen. These guys are going to break off. They're going to break off, and instead you're going to get a bond between carbon and nitrogen. And in fact, it's going to kick one of these hydrogens off in the process, right? So this is how you get this bond. So this red bond that we're seeing here between the two amino acids, that's how you get that bond. This is not considered a protein yet. This is just a string of amino acids. We call this string of amino acids a polypeptide. Oh no, I didn't realize it linked to the picture. Stop it. All right. In order for it to be considered a protein, it's going to have 4 parts. It's going to have the primary structure, the secondary structure, the tertiary structure, and the quaternary structure. The primary structure is the polypeptide backbone. Polypeptide chain. Right? That's what we just looked at. That's this structure here. The primary structure is the polypeptide backbone in an amino acid sequence. The way you can tell the polypeptide backbone is this repetition. We actually looked at this repetition in class. It's the repeated pattern of carbon, carbon, nitrogen, carbon, carbon, nitrogen, carbon, carbon, and then also on this side, nitrogen, carbon, carbon, nitrogen, carbon, carbon, nitrogen. This repeating pattern is going to be really helpful for you to be able to identify amino acids and identify how amino acids are going to bond together. Gosh, it always wants to take me to that picture. The secondary structure of an amino acid is based on hydrogen bonding. So what we have here is, if you look closely at this image, we've got let's do it in the red structure still, or the red picture still. You've got a amino acid here. You've got the R group attached there. You've got an amino acid C. You've got an amino acid there. This structure is going to hold its form because of the attraction of charge between oxygen and hydrogen. These hydrogen bonds that you're seeing are occurring because of the same thing that we saw with water. Hydrogen bonds where the oxygen, the negatively charged oxygen, is going to be attracted to the positively charged hydrogen. When it creates a spiral like this, when it creates this sort of twisted structure, we call this an alpha helix or an alpha helices in the plural. Helix is singular. An alpha helix is a structure of amino acids chunked together that then spiral around each other because of their hydrogen bond attraction. You can also get what are called beta pleated sheets, where you still get hydrogen bonding that's occurring, but because of the location where the amino acid is found, you're going to get a different structure. So the alpha helices and beta pleated sheets are formed based on where the amino acids, or what position the amino acids are in, right? So if you have, a lot of times people will draw amino acids as little circles linked together like this. The little circle just represents that whole structure that we've been looking at, amino acid. Now, each one of these amino acids is going to have either hydrogens or double bonded oxygens. This is a bad drawing, so don't take this to heart. They're going to have either hydrogens or double bonded oxygens, or their R groups sticking off, right? Now, these are going to potentially bond with, these are going to potentially bond with the opposite side or the same structure. If we had a hydrogen here, then the double bonded oxygens would bond there. Again, If you have a double bonded oxygen, then the hydrogen and the oxygen are going to be attracted to each other. So you get this sort of repeating pattern that can form either alpha helices or beta pleated sheets. And that's the secondary structure when we're talking about how do we make an amino acid or how do we make a polypeptide chain. This is how we do that via the secondary structure. The next thing we need to look at is the tertiary structure. The tertiary structure is further folding of the protein. So you might have an alpha helix here. You might have a beta pleated sheet here and an alpha helix. Oh, I just realized you guys can't see it. But you might have an alpha helix here, a beta pleated sheet here, and another alpha helix here. These are going to be held together by a number of different structures. These are going to be held together by a number of different structures. You can have hydrophobic interactions where these molecules are, again, like dissolves like or like attracts like. These two hydrophobic side chains are attracted to each other, and so they're going to want to stay close together. Keeping this, for example, were right here. This would be the attraction keeping this alpha helix and this beta pleated sheet close to each other. You can also have the same situation with hydrogen bonds. You can have this with ionic bonds as well, if you remember back to our chemistry discussion. But then you have these disulfide bridges. So this is where sulfur comes in, where that disulfide or that sulfide functional group that we were talking about, this is where that disulfide bridge comes in. The sulfur molecule is going to be attracted to itself and create a chemical bond that helps establish the structural integrity for the protein. Now, the final structure, the final thing that we're looking at when we're looking at amino acids is the quaternary structure. Now, not all proteins are going to have a quaternary structure, but complex proteins will. Quaternary structure is when you take multiple polypeptide chains. So remember, you're going to have alpha helices, beta pleated sheets, alpha helices for one structure. You're going to have alpha helices, beta pleated sheets, and more alpha helices for another structure. And then you can have even another structure. You're going to have alpha helices, beta pleated sheets, and alpha helices for another structure. And a quaternary structure is when all of these combine, all of these structures combine, and then they form a new functional unit. They form a unit that is completely different and has different characteristics than any of these structures were able to have on their own. A really good example of this is hemoglobin. So the molecule of protein in our blood cells that carries around the oxygen for us. So red blood cells transport oxygen in our bodies. The protein that transports oxygen in our bodies is going to have a very specific primary structure. Then that primary structure is going to form secondary and tertiary structures. We call this a beta subunit. Those secondary and tertiary structures are going to combine together with multiple other subunits. You're actually going to have two beta subunits and two alpha subunits. And then these subunits are going to carry oxygen in your red blood cells. So every one of these red blood cells is going to be filled with hemoglobin molecules, and these hemoglobin molecules all have the potential to carry oxygen, right? So this is an example of that quaternary structure. And this is just a summary of all of this process, right? You have the primary structure, which is the amino acid backbone, the amino acid sequence. You have the secondary structure, which is held together primarily by hydrogen bonds. You have the tertiary structure, which is held together by a whole bunch of other bonds like disulfide bridges, hydrophobic interactions, ionic bonds. And then you finally have the quaternary structure, which is multiple what we call subunits. Subunits are just individual proteins put together to have a new function that the subunits don't get to have on their own, right? Whew, we're done with amino acids. We have finished our discussion of macromolecules, and hopefully this was helpful for you guys.